Hello? Hello?
Hello,
Yeah, can you hear me?
am I audible?
Yes, yes, yes.
Yeah, sorry, I am late.
No, no, it's fine. You had work today or something?
Yeah, I was at work and just came.
Oh, okay. Fine. So, shall we start?
Yeah,
So, today, today I'll go through similar things. It will be assignments of week 7. Okay. So have you done this as assignments, or have you gone through the lectures?
Yeah, I have gone through the lectures of week 7 regarding the polymerizations and that part.
Mm-hmm. Yes. So we'll discuss that only today, and some assignments we'll try
Yeah,
to solve. So have you solved these assignments, or this is the first time?
yeah, I have solved this assignment. I
Okay.
have actually missed the week 6 assignment that I confirmed. I have solved the week 7 one.
Okay. So can you tell me how to solve this first question? What is
Yeah,
the
in
question?
this question, I was a little bit confused between option A and option D.
Okay. So.
Because I know that option A is the correct answer as optical energy is responsible for the process of getting cis to trans. But the question here is also that equilibrium statistics that also tells that when at the normal state there is a equal probability of having both the things means both cis state and trans state.
Hmm.
So, according to the question, when we look into it, the option D is also possible answer.
Hmm. Yeah.
So, I was just confused in between it.
And what is the
Uh,
correct answer
Maybe my
given?
understanding is not.
Option A is given, or which one is given? You can
Yeah, I
select any
have
also, right?
selected option A at the end,
Huh.
that is optical energy.
Huh.
Because of photon, the state gets transferred from trans to cis. But I was also confused that equilibrium statistics can be a thing because, as per
Hmm, hmm, hmm.
it has been asked that what is the primary effect.
Yeah, I think
So,
optical energy is more accurate
correct
than
answer.
equilibrium
Yeah,
statistics. Yeah, it will be like, if you have to choose between these two,
yeah,
optical energy will be the better choice. And if like multiple option correct, you can go with this, but it's fine. Optical energy is the correct answer. Fine.
okay. So, I have some misconceptions regarding the equilibrium statistics. That when talk about the equilibrium, then we are selecting a state.
Hmm.
So here, which state do we select exactly? Like which condition we will be considered as ideal for going like considering like all the molecules when are in the trans state, then there will be minimum probability that only few will be in the cis state. So
Hmm.
how are we considering the state
No,
exactly here?
at equilibrium, when all like both have not, like all the transfers have been stopped. Okay, so that state is the equilibrium state. So if there is a low
Okay.
energy and high energy difference, there will be exchange of energy, right? So
Yeah.
at it, at everything stops when the energies are equal. That is the equilibrium state.
Okay.
Hmm. So both the process happens there. Hmm.
Right, means the as soon as the molecule gets converted into cis, it again comes back to the trans state.
Yes, 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 yes. You are right.
Okay, that was just a misconception, so I was confused about that.
Okay. Fine now.
So here we can say that the same molecules jumps from one to one and one state to another state
Mm-hmm. Yes.
based on that timestamp or mean life. We are saying that after
Yeah.
its mean life is completed, it's again back into the cis state.
Yeah. There is equal probability of both the jumps.
Okay.
Fine.
Yeah.
So, what about this one? Question two.
Yeah, here when we say that which factors affect the steady strength.
Hmm. Yeah. So there.
So according to I have seen that I was confused a little bit here also that option A, that is dissociation rate,
Hmm.
that requires to be there.
Hmm. Yeah. Keep saying. Keep saying.
Yeah. So I was bit confused that association rate would not be that much affecting the factor, but dissociation rate would be a factor that affects, and monomer concentration is a great factor, because when we see that the higher the concentration is,
Hmm.
the higher the association rate is going, means they are in proportion with each other.
Hmm. Hmm.
And the length of polymer is also somewhat affecting, because if we start with a longer length of polymer, then we can see that association rate have a sudden, means have some increase, uh, because the crystal starts growing. Crystal is having a great surface area to grow.
Okay.
So when we choose a long chain of polymer,
Hmm.
the area increases. So we can say that the growth also increases with it.
So here, see the question says that what is the steady state length? Which factor affect the steady
Okay.
state length of the polymers based on a symmetric, both ends same growth model of linear polymers? So it's not static, right? The polymer is not static. It is growing, but in a steady state length. So that can
Yeah.
happen if it is getting expanded in this length in one direction and getting uh, dissociated from the other direction, right? Then the length stays constant.
Okay.
So that's what the question is about. See, see, first thing is steady state length of the polymer based on symmetric, both ends the same growth, symmetric growth. So that this can only happen the steady state, like if one end grows faster than the other, then it elongates, and if one,
Okay, so I have actually understood it wrongly, that I have considered the steady state growth, means I have considered the rate to be staying steady.
okay, okay, no, so
So
that's,
that way.
yeah, this is there was a misconception here and got it. So dissociation rate and association rate both are important, and monomer concentration is also important, but length of the polymer may not be the most accurate thing because length becomes fixed.
Okay.
Yeah. So
Uh,
first three will be the correct, more accurate options.
okay.
Okay. Sorry. Yeah. So question three. Have you solved this? Which of the following corresponds to features of the nucleation dependent polymerization model of cytoskeleton system? Multiple options shall be correct. So the nucleation dependent polymerization. Nucleation
Yeah,
dependent polymerization.
dependent systems.
Do you know what is this three phase behavior?
Yeah, three-phase behavior is when the molecule is also in all the states. Means it is showing behavior for all the three states.
All the three states means
Sorry, three phases, right?
three phase. Yes. So what are the three phases? Any
Yeah, that is. Okay.
so okay, I'll do it here. I need a blank page. So yeah. So the three phases are, so what's the question? Let's, so nucleation dependent polymerization happens. So there are three major phases. One is lag phase, one is growth phase, and one is plateau phase. So lag phase, growth phase, and plateau phase. So lag phase where there is nothing much happening, then there is the growth, and then it becomes a plateau. Okay, so these are the three phases. This kind of uh, plot
Yeah.
you can have, right? So this is how polymerization dynamics has three phases. Now if you like plot polymerized state versus time, you will see three phases, right? So this will be correct answer. Next is uh, the addition of small oligomers that serve in nucleate the growth do not alter kinetics. Is it correct?
Mhm. Yeah, the small addition.
I
No,
think
they alter the kinetics. When we add the small oligomers, they alter.
they alter.
So
So
that is not correct.
this is not correct. Yeah. And next is the slope of the increasing polymerization can be the slope. The slope can be considered to the estimation of polymerization rate. This is also correct. Why?
Yeah, as
So
we see in the graph.
yeah. So the slope is the quantification of the rate of polymerization.
Okay.
Yeah. So suppose this is how we quantify rate, right? Rate of, let's say, rate of displacement. This is called velocity, right? So
Yeah.
so this is how this quantification is happening. The steady state absorbance in equilibrium phase changes depending on whether perform perform nuclei have been added or not. Yes, this is also correct.
Yeah, so this is also correct. Yeah.
So A, C, and D, all three are correct. Okay.
Yeah.
So next question is filament treadmilling requires multiple conditions to be satisfied. Choose all the correct conditions. So what is filament treadmilling?
So the treadmill is when one a filament is continuously growing and also decreasing from one side. Like it is growing in one direction and suddenly decreasing from other direction like
Yes,
this.
yes, correct. So there is like uh, growth like this, like a treadmill, like it goes like this, right? So this can
Yeah.
happen via asymmetry in growth rates at two ends, right?
Yeah, that's correct.
Yes. Transport of monomers. So what happens is this: there is no transport of monomers happening here in filament treadmilling. There is like monomer does not move from here to there. Like there is no transport happening. So this is not the correct answer. At the treadmilling critical
Okay.
concentration, the rate of monomer addition at one end equals to the rate of monomer removal at the one end. Yes. So this is also
This
correct.
is also partially correct, no?
Why partially?
Because when we consider the rate of addition, so it should be asymmetric. From one end it should be more and other should be less.
Yes. But you're saying that at the treadmilling critical concentration. So there is a, like the whole phenomena is
Okay.
growing like this, but at critical concentration is defined like it is the, like you can say, the junction point. Like after that the treadmilling happens. So this is the critical point where the both at the both the ends the addition and
Yeah.
subtraction are the same rate. So this is also correct. Yeah, and this is also quite intuitive. Neither end grows or shrinks.
Yes, so it is also correct because it's in another line written in another word
Exactly,
meaning.
exactly. Simple, simple terms.
So just one question.
Hmm.
Are the same kind of questions in that exam or they would be something else from the syllabus because sir have taught many more things in the
Yeah,
lecture.
yeah.
Out of that, only few questions are given in the assignment. It's only a few kind of subjects are touched.
Yes.
So the exam paper would be also in the similar manner, or it would be from the also from other sections which are not touched, which are there in the lecture but not given in the assignments.
Yeah, so I can't exactly tell you this. So the thing is,
Okay.
actually, I don't even know about the question. So what I can say is, if there will be emphasis on these exact points which have been in the assignments. So do them properly, and just don't leave anything, just try to have some idea. Okay, so
Yeah, that is asking that all the equations out of only we can find only few equations to be there in the a few type of equations are being asked in a question form in the assignment. But in class there are many topics and much more knowledge is given than that has been asked in the assignment. That is why
yes,
I just clarify for the that how are the other subjects are being asked. Like what are the other subjects being can be framed as a question. That was my point because I cannot found any such previous year papers or that for reference purpose.
yeah, there is no such things like previous year, so there will be emphasis like, on these topics, but do this properly, and rest you can like have, you
Mm.
can choose like which themes to be presented, or from which topics there can be more problems, numerical problems in that. Yeah, like you
Yeah, I
can
will
take a
go through
call.
all this.
Yeah, take a call. Mm. So next is question 5. So part M is a type of bacterial cytoskeleton system. Choose correct option that describe the dynamics of the physiological role. So what is part M? It's a type of bacterial cytoskeletal system. What is cytoskeletal? You know what is cytoskeletal?
Yeah, that is something which the insects have, like ants, crabs, and they have something which is called a cytoskeletal system, which is protecting their outer body. Like we humans have skin, they have a skin, but they have also had extra protection.
Yeah, it's like
So it's
skeleton.
like a cytoskeleton.
Skeleton, you know, skeleton. We also have skeleton, right?
Yeah,
So for them, it is called the like, same kind of uh, job it has for bacteria and cells, which is called cytoskeleton. It's the same effect. It protects the whole structure. It gives the structure of the body and protects from any kind of damage.
steady. Yeah.
Yeah. So homolog of actin. So it's like an actin. This first option will be correct. Parent. So it's like it looks like actin only. So that's why the
Yeah,
homolog
that is
term.
correct. Estine is there in crabs and the insects. So
Yes.
that is correct. It's homolog.
So polymerization dynamics of parem are involved in genome segregation of bacteria.
Yeah, this is also correct.
Yes, this is also correct. The behavior of parem cannot be explained by simple nucleation dependent polymerization model. This is also correct. So simple nucleation dependent, it cannot be. There are more things are happening there. Polymerization of parem is involved in the segregation of low copy number plasmids. This is also correct actually.
Right, okay, all
All
four are
all
correct.
four are correct. Yes. So week 7 last one, which is the which of the following variables can determine the equilibrium polymer distributed distribution of steady state polymer. So do you know what uh, this model is about? PN model, cytoskeletal polymerization model.
Yeah, that is about the strength over the force applied.
Hmm. So what does this PN steady state PN tells you? There in the lectures he has covered the this calculation. Do you remember how was it? Like how did he calculate?
Might be, I forgot.
So okay, just give you some hints. Hints means try to give you some idea.
Yeah, I remember.
So this structure you remember, right? So there is dissociation happening k at a rate k off, and association can happen at a rate k on from this end also. Also from this end there is k off dissociation rate and association rate k on can happen, right? Now
Yeah.
we define Pn at T. So what we define Pn at T? This is polymer of length n in monomer units at time t. So this is essentially a length of polymer which suggests that it has n numbers of monomers. In monomer units means it has n numbers of monomer in the polymer. Okay, at particular time t. Fine.
Yeah.
Okay. Now at equilibrium, Pn plus P1 is equivalent to Pn plus 1 with a rest constant Kd. Which what does it mean? Like if you add one more extra monomer there, this is P1, you get Pn plus 1, and at equilibrium this Pn and P1 and Pn plus 1 are interchangeable. So that's the definition of equilibrium, right?
Yeah, there are both the states at same time.
Yes.
The
So
probability of going from one state to another state is equal.
exactly.
So the association is also happening in the same time, and dissociation is also happening with the same rate.
Yes, yes, exactly. So we can define this rate to be the ratio of this two. Yes,
Yeah.
yes. So this is how we define the rate. Now, what we will do, we'll try to calculate the. Uh, so now we come back to this question. Now, what is said, equilibrium length distribution steady state FPN, and this length, what does it depend on? Of for polymers of length n. So concentration of monomers at P1. So it depends on concentration of monomers, right?
Yeah.
Yes. And dissociation constant of monomers Kd, it depends on this, right? Kd is there.
We have seen.
Number of initial nucleating oligomer added. So whatever the number of initial oligomer, it will depend on this also.
But size of monomer does not matter.
Does not matter, exactly. So individual size of monomer, sorry, individual size of monomer does not
It
matter.
does not.
Okay, so this was this week 7 questions. So one more topic I will try to give some hint about week 8. So actually, mm the, this was supposed to be the week 8 last week, but we are lagging behind
Yeah.
uh, 1 week. And for the last week, I was planning to do about this Python programming thing. So which
Okay.
I don't think you will need, right? It will be
Yeah,
pretty
I don't need that.
it will be pretty basic Python programming, and I will like it will be more or less how to solve equations and how to plot certain stuff about that. So what I will do, since you are the only one joining, I will not have any other lectures. I will record the whole video and upload it to YouTube. That I was planning. Okay. So
Okay.
if you don't need, you don't have to come and join. I think that will, if anyone wants it, this like this detailed about Python programming and how to do scientific calculation there, I'll think I'll cover those in those videos, in that video. Okay. So one last
Yeah.
thing, I will just give you some hints. It is week 8. So have you gone through week 8 lectures?
Yeah, I have just started, but not completed yet. I have just seen lecture one that is about the symmetricness of the which we can see in the
Membrane.
plants.
Membrane, right?
Phyloelectric axis, yeah.
Yeah. So there are membrane
That is
dynamics.
about the patterns that we are following in the nature regarding the geometry and all those things.
Yes. So there are membrane dynamic. There is Turing pattern, and there is phyllotaxis.
Yeah.
So it's mostly, all of them are mostly about patterns. So phyllotaxis, this flowers and all have this kind of pattern
Structure, proper
structure.
structure.
Yeah. And Turing pattern are like talks about how this uh, tigers, zebras, they get the skin pattern and all. And membrane dynamics, you know how a membrane grows and stuff. So I'll just give you some hints about Turing pattern. So do you know about Turing? Who is Turing? I think
Yeah, Alan Turing.
yes, this is Alan Turing, the same one.
Yeah, the father of computation theory and also
Exactly,
the father of AI. We can
exactly.
say AI theory is
The
being defined by Alan Turing.
imitation game, right?
Yeah, Church Thesis, his most famous paper.
Yeah, yeah. His most famous papers were there. There he wrote another paper about
Yeah, that is
biology. So about
tuning patterns.
yes, exactly about biology, and this is like crazy famous in this. All the
Yeah.
like mathematical biologists, the callers the paper called like chemical basis of morphogenesis. The paper's title was
Okay.
chemical basis of morphogenesis. So morphogenesis means the study of shapes and patterns. So I think Alan Turing, I forgot the year, 80s, 80s or something, I forgot. So you can look at it. It's pretty famous paper where he suggests that you can write some equations if there are competing concentration. It can be any kind of concentration. He in his paper he talks about chemical concentrations of certain chemicals in a biological system,
Yeah.
but it can be any kind of concentrations, does not matter if they are coupled via some functions some functions, then with specific choice of parameters you can achieve these kind of patterns, all these kind of patterns. So what do I mean by that? Look at these equations. So what do you like, what do you see in these equations? So let's say
These are the partial derivatives.
yes, let's say we don't have this part, okay,
Difference.
let's say only this part. So what does this part tells you? Let's say there is no this part, only this. Do you, does this equation looks familiar to you?
Yeah, it's derivation, second degree derivation.
Yes, it's this Laplacian is suggests del square del x square plus del square del y square plus del square del z square, right? So this equation is called diffusion equation.
Okay.
So do you remember we talked about random walk?
Diffusion.
Yes. So we talked about random walk like this. Remember we were
Yeah.
talking about that day. Now this is microscopic model of random walk. Let's say you have now for macroscopic model you do the same thing with you say that you spray some perfume here in this room, and the concentration at this position is, let's say this is let's say you have axis x and y axis, right? I have to do it properly. We need a blank paper. Okay, let's do it here. So you remember random walk, we talked about perfume spraying in the whole room, right?
Yeah, it gets distributed over the whole area.
Yes, like this.
Because of the kinetic energy, it's been got.
Yes, so all of them are doing this kind of random motion. Now in a continuum setup, sorry, so in continuum setup, what you do is, it what does it mean? It means you don't care about individual molecules anymore. You care about the what is the density of the,
Hmm.
let's say, perfume across the room. Okay, so at t equal to 0, what is the density? Let's say this is C. So what is the density here? It's 0, right? Here.
So it's yeah.
So everywhere it is 0 at t equal to 0, only in a particular area. Let's say this is xy and you have this x0 y0. Now see x0 y0 t equals 0 is, let's say, c0, and this xyt equals 0 for x not equals x0, y not equals y0. Do you understand these two lines?
Yeah. There is some area where the concentration is high and all other places the concentration is zero.
Yes. Now, if you can say, if you can find what this cxy at t equal to some t, let's say at some t, not 0, but t non t greater than 0.
Yeah.
If you can find it, then you can tell how this concentration field, how this perfume concentration is evolving or spreading over time, right? Is it fine? Hello?
Yeah.
Do you understand what we are trying to do? So, you know that
Yeah,
the.
we are
Huh.
actually
Yes? Come again?
sorry, it seems some network problem, voice is lagging or
Oh, acha. Could be. Actually, like there has been some kind of. Like, in my place, the network is fluctuating a lot. Yeah. So, what I'm saying that if you can solve, if you can calculate concentration at any xy at any t, then you know uh, how the concentration is evolving with time and how it is spreading over space, right?
sorry, but I just I can reconnect.
Yes,
It
try.
seems that voice is lagging in. I just reconnect.
Okay, okay.
Hello, am I audible now?
Yes, yes, you are audible. I think there is some issue with my internet. So this is what we are trying to do.
Okay.
So what I'm trying to say, sorry. So what I'm trying to say, if you have cxyt, where x is any position in x, y is any position in y, and t is the any time. If you know that, then you will know how the concentration is evolving over time and how it is spreading over space. Right?
Yeah, by finding t at different time instances, we can actually calculate this.
Exactly. Exactly. So this is same problem. This is the equivalent problem of Brownian motion. Sorry. So in Brownian
Yeah.
motion, we look at every particle and how they are moving and where they are going. But in continuum model, in diffusion, this is called diffusion model. This is diffusion. In diffusion, we don't look at every particle. We look at the overall concentration over the space and time. Fine?
So we are actually looking over all the concentration that are present in one specific area, means a limit of
Not
area,
not
and then we are seeing that what are the different concentration at different points.
yes, yes, yes. So initially this is the initial, and eventually it spreads out, right? Like this, somehow it
Yeah.
spreads out. Now, if we can find out this quantity, you will know for any time I ask you what is the concentration. You will know for in any position I ask you, you can tell me what is the concentration. That's the exact same thing
Yeah,
in
we can know the concentration in any time based on what is the density at that area in that at that point means if
Yeah,
we
yeah,
consider a
concentration means
proper area then we can actually calculate the density for that.
yes, it's meant density also. Yes, it's the same thing. Now, the mathematical equation that describes the evolution of this C is, so let me write it down. Mathematical equation that describes the evolution of CXT, sorry, CXYT is, this is called the diffusion equation. What does it look like? Change in C with respect to time is equals some constant times, let me write it like this. So this is how it looks. This is how it evolves. So what does it tell you? Can you interpret it? Try to interpret it. So the left hand side, what is partial? This is del t. You know what is this?
Yeah, it's differentiation of any rate of it. It's showing that if we are getting a continuous curve, then we need to find a specific amount of rate. That at what rate the curve is getting either smaller or bigger. So we need to calculate it. So we are actually differentiating it in a different paths
So what is the
to get a
different?
smaller unit based on the limits what we choose.
So what is the difference between this? Not t. Sorry. Let's write x.
So, partial differentiation is again related to another variable which is also not constant, but differentiation is related to constant variable. So means differentiation is related to only one variable, but partial differentiation is related to two or more variables.
Let's say let's say if you have a function of x comma y with which looks like 2x square y plus 5. So what is df dx? Tell me. It's, can you tell me what is df dx and what is del f del x?
Yeah, that is 4x.
Which one? Is this 4x or is this 4x? Which one?
Sorry?
4x. Which one will be 4x? This one or this one?
Yeah, the d by dx would be 4x because we can consider y as a constant here because we are not considering it to be a variable because we are dealing with only x. That is differentiation.
So will there
But
be any
when we
y there?
consider
4x y or 4x? Is it 4x or 4xy?
it would be 4x only because we are considering differentiation in regards to x. So we are considering y to be a constant. So it would be 4xy.
So 4xy will be there, right?
Yeah, yeah, not 4x only.
Yes. So you have some a little misconception. Whatever you said, it is true for this one, partial. So the term partial means you don't do derivative of the all the variables. You do only for the variable given. That's a partial derivative. For this one,
Okay.
df dx, you don't consider y as a independent variable or constant. You consider y as a dependent variable. So what the answer will be? You first 4xy plus,
Plus dx by dy.
yes, this will be the
Yeah,
answer.
dy by dt.
Yes, fine. So again, now you understand this all these meanings, right? So del c del t. So c depends on x, y and t. Now when you do a derivative with t, you consider x and y as a constant. Next is del square del x square, like it is the double derivative, you do it twice. So you look at this equation. This equation tells you, if you can solve this equation, it tells you how c, the variable c, is evolving with, how the variable c is evolving with time. So if you solve this, you will find the whole profile of c. So for one very famous example is heat diffusion. Do you know about heat diffusion? Wonderful.
Yeah.
So what happens is, let's say you have a rod like this. Let's say 1D rod. Okay. Now at t equals 0, you heat it up here. Now you ask, what will be the temperature of other places at some t? Like what will be
It
the
will soon get to more hotter and hotter on the side which we are eating. So
no
it is going in a,
no
we
no we
can
are
say, in
not,
a line which is propagating.
see, we are not heating it. We just say that we have heated it up to t0 here, and we stopped
Yeah.
heating. Now what will be the temperature at t equal to 0 here? It is 0, right? At t equals 0.
Because we are not.
Now you are asked that what will be the temperature here at t equals some t naught, which is greater than 0 at some point. This also can be solved via diffusion equation.
Okay.
This is 1D, that's why so heat also diffuses. So at t equals 0, if you want us to plot it, it will look like this, right?
Yeah.
Now if you solve this equation, I am not going into the details of solving this equation because it takes uh, algebra a lot, different algebra, and which is like I have to explain a lot more things than
Yeah, I should probably brush up my 11 and 12 concepts.
yes,
So
yes,
I
because
will do
so
it.
these are not, see, if for these exams and all, these will not be necessary. I'm just giving you the idea what is the diffusion equation is. Okay. So it's good to know so that you can understand Turing's equation a little bit more, but it's not that important. I'm just saying that if you solve this, like I'm just giving you the feeling that if you actually can solve the, what will be the profile of this heat or any concentration that is doing following this kind of equation. So if you solve this, it eventually becomes like this. So at, let's say, t equals t1, the more you wait, you see this peak reduces and becomes like this. So this is t equals t2, where t2 greater than t1. So this peak temperature here reduces and all the other increases. Here the temperature reduces, it was here, from here it, like let's say it was here, the initial temperature was here, and it reduces to this and others are increasing. So this also comes from this heat equation. Okay.
Okay.
So the, and if you solve this equation and if you do the random walk motion, you will get exactly same result at, like at some x, y and t, what is the concentration of the perfume, you will get the same result. So this is called diffusion equation. Fine.
Yeah, so because of that concentration, we are getting the Turing equation or Turing patterns.
Now Turing is saying that, okay, now you have two different kinds of concentration, one is u and one is v. Okay. So one is u and another one is v. Now they itself are doing this diffusion over space. Naturally they are diffusing over space. Now let's say, on top of that, they are in a interacting, they are also interacting some chemical, let's say they are chemicals. What happens, chemicals interact and they change, so there is some functional form involved which are affecting, like one concentration is affecting another, another one is affecting this one. If you have this kind of form, now like for example, if uv can be like, let's say u square v plus 2u anything, right? guv can have some kind of from uv square plus 2v, this kind, this kind of form with some constant involved, k1 k2 k3 k4. Now he is showing that this, depending on this du, this du dv k1 k2 k3 k4, this constant, if you play around with this constant, you can get any kind of this kind of patterns. His argument was that for the leopard or cheetah or zebra, the underlying process of the chemical concentrations that are there, that are caused, that are causing the patterns, internal process is same. By species to species, these values of these constants are getting changed, and that's how different patterns are evolving, arriving. So what
Okay.
the importance of Turing's work is, he came up with a mathematical model, a mathematical mechanism that that can predict very precisely what will be the pattern formed in any real biological species, right?
Based on the concentration in different
Concentration and
times when we measure
yes,
it.
yes, concentration and how they are interacting with each other.
Reacting
This
each
fu
other.
and f and g are important. Without this you do not get any concentration.
Yeah.
So that is there, you so I can go through the lectures he has talked in one lecture in about Turing pattern. And lastly he did this phyllotaxis, where like there are three lectures on that,
Yeah,
about
I
the,
have first started with the pilot exercise.
yeah. So go through them. It phyllotaxis is not that difficult, it's easy. So that will be
Yeah, that is why I have started it first and then I will jump to more bit complex topics. Yeah, I will go through it.
yeah, 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 definitely. So that will be it from my side. I will just one more that Python programming, and I'll try to solve this kind of
Okay.
equation and plot this. You can watch it over YouTube if you feel like, or you can just skip it, no problem. Okay.
Yeah, so if it is on YouTube, then there is some approximate time or which.
When you will get it, you are asking.
Yeah, yeah.
Okay, I'll try to do it soon.
Okay.
Like by next
Yeah.
week definitely you'll get it, but like I'll try as quickly as
So
possible.
also if it's on Monday or Tuesday, then if I can join.
It's fine, I'll let you know if it's so
Okay,
I'll just
okay.
go through, like you have seen that there are three lectures on Python programming.
Yeah.
Yeah, so that I'll try to give some hints, like about uh, like there are some students who are not familiar with coding that much, so
That is, yeah. Okay.
so okay then, nice meeting you.
Okay.
Yeah, good luck for your exam.
Thank you.
Mm. Thank you. Bye.
Thank you for teaching this much time.
Yeah, it's fine. Yeah, yeah, hope you liked it.
Yeah. I actually got many things cleared which were not being clear. Like the equation part was, I never thought it that way.
Which one? Which one?
The discussion equations, yeah, I never thought about solving them and relating, correlating them with physics in that way.
Ah, I see,
When
I
I
see.
studied the maths, yeah,
Ah,
that I never did it. So
ah.
this is the thing which I actually love,
Yeah, this is really
correlating the
interesting.
equations with physics. Yeah.
Yeah, that's what we do, right? Like we means I personally, I do that. I study some biological system. I write some equation and solve it. And then I predict what if you do some experiment, what will you get? And some other guy from some other lab does the experiment. I don't do it. I just solve equations. Yeah. Yeah, that's nice. And thanks.
Thank you, sir,
Yeah,
for
sure.
teaching this. Yeah.
Yeah. Okay. See you. Bye.
Okay. Bye.
Bye.
If you look for this book and the course related to it by Philip Nelson, Physical Models of Living Systems, it's kind of a biophysics by programming book. Okay, and it has similar ideas that I have discussed in class about randomness and random walks, and it has some coding examples that you should be able to find. All right. So let me see. We have some program code. So in fact, what you're asking is a separate course. I know that NPTEL also has some Python courses, but I would recommend you at least look in your library. Are you a biomedical sciences student? Is PGI familiar with the Postgraduate Institute of Medical Sciences, right?
Yes, I'm doing my medical biotechnology there.
Okay.
Master's degree.